Hi, everybody, and welcome back to this lecture. What we're going to do is we're going to pick up where we left off last time, and we're going to go ahead and take a look at what glycolysis is going to be. Now, this is going to be our first step in cellular respiration. So what we're going to try to do for this lecture is we're going to try to identify exactly where glycolysis is going to happen. We're going to look at the different steps for this portion, and then we want to figure out exactly what are we making at the very end. So let's go ahead and get started and see exactly what glycolysis is in respect to cellular respiration. So the first thing I'd like to do is I want to break down exactly what glycolysis means, or in this case, what parts are they kind of trying to tell us. So when it says glycolysis, what essentially that means is glyco is going to represent sugars, and then lyse is going to be breaking apart. So in this case, this step is solely responsible in taking in glucose and breaking it apart in order to get something known as our pyruvates. So glycolysis is going to be the first step in breaking down glucose, and this is going to happen in the cytoplasm. So you can see this is our cell right over here. We know that the mitochondria is going to produce a lot of ATP. In this case, it's making its way to the mitochondria, but it's not there just yet. So cytoplasm is going to be where glycolysis is going to take place. Now, what's essentially happening here is we're taking one glucose, which is going to contain 6 carbons, and we're going to break these down into 2 pyruvates, which is going to be 3 carbons. So in this case, we're taking our glucose and we're going to have 1, 2, 3, 4, 5, 6 carbons, and essentially we're going to lyse again or break them apart from our 6 to our 3 and our 3, in this case producing those 2 pyruvates. Now, the thing that's really important about this is as we're converting glucose into our pyruvates, we're going to actually produce 2 very important molecules. In this case, we're producing 2 ATPs, and we're also producing 2 NADHs. Now, these are going to play an important part in the next coming slides. But again, just know that we're taking our glucose, which is our 6 carbon, lysing again or breaking them apart, and we're producing 2 pyruvates which contain 3 carbons. As we're doing that, we're making 2 ATP as well as 2 NADHs. Now, in glycolysis, this does not require oxygen just yet. So again, oxygen is not going to play a part in glycolysis. So let's go ahead and see exactly what that all means. So this is going to be a diagram showing us exactly what's happening in glycolysis. So again, we have our glucose and we have our 1, 2, 3, 4, 5, and 6 carbons. Now, as we're lysing, remember breaking this apart because we want 1, 2, 3 carbons, which is going to be our pyruvates, which you can see here, 1 and 2. So as we're lysing or breaking this apart, what's going to happen is we're going to take our ADP, adenosine diphosphate, and it's going to go ahead and get that extra phosphate group as we're breaking this apart, and we're going to be producing 2 ATP. Remember, ATP is that very energy dense molecule that we want. Think about it again like a battery. ADP is very, very energy inefficient. It doesn't have a lot of it. And then as we're adding that extra phosphate, now we're storing or charging up that energy or that battery. Same thing over here. We're going to have 2 NAD pluses, but as we're producing these 2 pyruvates, it's going to go ahead and go through this. It's going to pick on an electron, and it's going to become NADH. So again, we're going to go and produce our 1 and our 2 pyruvates, and we're also going to produce some heat. So in this case, our input is going to be 1 glucose, and our output, what we're producing, is now 2 pyruvates, 2 ATPs, and 2 NADHs. So again, pyruvates are going to be used in the next step of cellular respiration, and NADH is an electron carrier represented by this E, and will be used in the final steps of our cellular respiration. So think about this as precursors or kind of prep work or setting up for the other steps of cellular respiration. So one thing we kind of really need to understand is what's going to happen when we don't have oxygen or we have low or limited oxygen in the mitochondria. Remember, in glycolysis, we're not utilizing oxygen just yet, but oxygen is going to play an incredibly important part when it comes to producing ATP. So again, when the mitochondria are limited or O2 levels are low, hypoxia, remember low oxygen, the ability of the Krebs cycle and the electron transport chain to produce ATP is going to be severely limited. Okay, So again, in our glycolysis, remember this does not require oxygen just yet. We're taking our glucose, 1, 2, 3, 4, 5, 6 carbons, and we're going to lyse them, break them apart, going through glycolysis, producing 2 ATP as well as 2 NADH plus, oh, I'm sorry, NADHs. And then we're going to end up with our 2 pyruvates. So 1, 2, 3, 1, 2, 3. And in the absence of oxygen, or in this case, when oxygen levels are very low, the mitochondria can't produce enough ATP. So it is not going to take this pathway, but rather go through lactic acid fermentation. Okay? So lactic acid fermentation is used by the muscle cells during high intensity activities or when they are fatigued and by the red blood cells, which lack mitochondria. So this is what that's going to look like. So in anaerobic respiration, again, we're going to have 2 steps, just like our previous ones. Step 1, glycolysis. We're still splitting apart that glucose into 2 pyruvate molecules. Glycolysis, again, is still going to produce that 2 ATP and 2 NADH. Again, this does not require our O2, so this isn't going to be necessarily really effective. And then step 2, NAD plus regeneration. Pyruvic acid is converted to lactic acid. So remember, this is the part where there isn't enough O2, and it's going to take this pathway instead of the other normal pathway in order to produce ATP. So in this case, this step regenerates 2 NAD plus molecules, which are used in glycolysis. So this is what that's going to look like. We have our inputs here. We have 1 glucose, and we're going to end up with 2 lactic acid and 2 ATPs. So unlike this part over here, where this is going to be regular glycolysis, this is what we want. This is the precursor, step 1, to the circular respiration, because in step 2, we're going to the Krebs, and then step 3, we're going into the electron transport chain. So again, there's only 2 parts to glycolysis. 1, taking glucose, so step 1, and then we're going to go ahead and lyse that, break that apart, get 2 ADP, 2 ATP. I'm sorry, we're going to take 2 ADP and produce 2 ATPs, and then we're going to take 2 NAD pluses and create 2 NADHs. And then in part 2, we're taking our pyruvic acids. And again, they're going to be used in the next steps for cellular respiration. So the outputs for regular glycolysis going to the Krebs cycle is 1 glucose, and our output is 2 pyruvic acids, and then 2 ATPs and 2 NADHs. But in this case, when we're looking at the absence of oxygen, again, it's going to go through lactic acid fermentation. So in step 1, we're taking that glucose, we're lysing it still. We're still going to produce 2 pyruvic acids. We're still making 2 ADP and 2 ATPs. And again, we're still producing our NADHs from our NAD plus. But again, because we don't have oxygen, right, the absence of that, it's not going to go to the Krebs, and it's not going to go to the electron transport chain. Instead, it's going to go through fermentation, and it's going to be producing lactic acid right here. Now, when we're going through fermentation, that's not always necessarily going to be a bad thing. In fermentation, we often use this in order to produce various products, including alcohol, beverages like beer and wine. We also use it to produce things like soy sauce. We also use it to produce things like cheeses and yogurts, beer, wine, and bread. So again, we can utilize anaerobic respiration in order to produce a very wide range of products by just removing or introducing a very low amount of oxygen and allowing it to go through that fermentation. All right, and that is going to conclude the glycolysis, or step 1, glycolysis. Again, the main takeaway from this is you're just taking glucose, you're breaking it apart, and you're just making 2 pyruvic acids. We're also going to be producing 2 ATPs along with NADHs. So take a look at these review questions, see if you can answer them. I'll give everybody a couple seconds to pause this, give it a try, and then we'll look at it together as a class. All right, welcome back, and this is what you should have gotten. So The energy currency used by cells is going to be ATP. Remember, ATP is going to be very energy dense. ADP is going to be very energy inefficient. It's not going to have a lot of energy. AMP is something we did not talk about. And then adenosine is just going to be one portion of ATP. So in this case, ATP is going to be the answer. Again, it's going to be very energy dense. Which of the following are products of cellular respiration? Remember, we're producing ATP and we're also releasing some carbon dioxide. Where does glycolysis take place? It is not going to take place in the cell membrane, not in the nucleus, not in the mitochondria. Although there are going to be two steps that do take place in the mitochondria, glycolysis specifically is not going to be in the mitochondria. It is going to be found in the cytoplasm. The glucose that enters glycolysis is split into two molecules of what? It is not going to be NADH. Although this is going to be a product and we are producing two of them, it is not going to be NADH. Again, we are going to be producing to ATP from glycolysis, but again, this is not going to be those two molecules that are going to be a result from glucose. NAD plus, again, is not. That is going to be something that's waiting for that split to happen in order to pick up some energy to become NADH. In this case, it is going to be our pyruvic acids. Remember, glucose is going to contain 1, 2, 3, 4, 5, 6 carbons, and pyruvates are going to be 1, 2, 3. So the splitting of this is going to produce 2 ATP along with 2 NADHs. Glycolysis produces two molecules of, in this case, ATP, and two molecules of NADH. It is going to be this right here. What is the final product of anaerobic respiration? So in this case, it's going to be the absence of oxygen. We are not going to have that. So remember, we're going to go through glycolysis first. And instead of going to the regular route with the Krebs cycle and the electron transport chain, because we don't have oxygen, it is going to go through lactic acid fermentation. Finally, which of the following is not true of glycolysis? It is going to produce 4 net ATP. Remember, in this case, we're only producing 2 ATP from that split. All right, and that is going to conclude this lecture. So as always, if you do have any questions, comments, or concerns, please feel free to reach out and ask, and I will see everybody in the next one.